We're back for the third part of cells. We're picking up with the membrane again. We were talking about the different types of solutions. The first thing that I want to point out is that when we're considering these solutions, the terms isotonic, hypertonic, and hypotonic are comparison terminology. What I mean by that is that there are two solutions that are being compared. In life, we are comparing the solution that is outside the cell to the interior of the cell. Remember that the interior is an aqueous water-based solution. If you say that we have an isotonic solution, it means that the solution outside of the cell has the same concentration of solutes, the stuff dissolved, as the inside of the cell. The net effect of that scenario is there will be no overall change, as many molecules of water will move inside of the cell as those that move outside of the cell. Everything pretty much stays the same. In a hypertonic solution, what we're saying is that the outside of the cell has a higher concentration of solutes. More dissolved stuff exists outside of the cell than inside. Remember, we're comparing two solutions, so the outside has to be hypertonic. The outside is hypertonic while the inside is hypotonic, meaning having a lower concentration of solutes. Water always moves towards the hypertonic solution, hyper meaning higher. So, a higher concentration of solutes is sucking the water to that side of the membrane. Solutes suck water. A simple phrase that might help you remember what is occurring when faced with some scenarios. Water will move to the hypertonic solution. That means that the cell itself is going to shrivel up because the cell is losing water. A hypotonic solution is referring to the fact that the outside of the cell, there's less dissolved solutes compared to the inside of the cell. There are more dissolved solutes inside of the cell. Remember, if there's more dissolved solutes, the concentration of water must be less. So water is going to move into the cell because the cell has more dissolved solutes than the outside of the cell. Solutes suck water. So water is moving inside of the cell. If the cell does not have a cell wall, then the cell membrane is going to expand and it can burst unless the cell is designed to withstand this. An amoeba, which lacks a cell wall and lives in a hypotonic environment such as pond water, will have a special organelle that can pump the water out. If it's a cell like our red blood cells, it does not have the ability to deal with water that's moving in. The red blood cell in a hypotonic environment will experience the cell membrane as swelling as the interior expands with the incoming water. The membrane can break. A plant cell or a bacterial cell that has a cell wall is not going to break in a hypotonic environment because the cell wall will constrain the expansion and prevent more water from coming in than the cell can withstand. In sum, water will move into the cell if it's placed into a hypotonic solution, but the cell wall will prevent the membrane from expanding any further than the barrier of the cell wall, and therefore the cell will be okay. If a cell without a cell wall and without special organelles is placed in a hypotonic environment, it can swell and break. I hope that you can work to understand these different terms and what will happen to the cell in the different scenarios. Make sure you feel okay about this concept. This is something you will be asked about in the future. We're next going to move into the processes that require energy to cross the membrane. We'll talk about these different systems, but don't spend too much time fretting over them. I really want you to focus on just the fact that we're going to need energy for this to happen, and that there are different systems that the cells can use to get what they need across the membrane. You can study these in much more detail in a cellular and molecular class or a biochemistry class. This figure from your book is illustrating the different types of systems. Uniport is referring to one type of molecule is going to cross the membrane with this type of protein channel. In order to cross, the cell is going to have to expend energy. This is because going against the gradient requires energy. Remember, with passive processes like diffusion and facilitated diffusion, Molecules are flowing along the gradient. They move from areas of higher concentration to lower concentration with passive processes. With active processes, energy is expended to move molecules from areas of low concentration to areas of higher concentration against the gradient. Consider something like glucose. The cell really wants to bring it inside. If there's more glucose inside the cell than outside of the cell, it's going to get that molecule across the cell by expending energy and using a specific channel protein transporter. Where does that energy come from? ATP, of course. Many students are already familiar with ATP as an energy source. We've looked at it in the chemistry section. Adenosine triphosphate, ATP, which is an RNA nucleotide. It's nothing magical. The cell will clip off a phosphate from ATP, leaving you with the diphosphate, ADP. This phosphate usually gets removed from ATP and then stuck onto some other molecule, at least temporarily. So the ATP is utilized to obtain energy needed to move a molecule across the membrane against the concentration gradient. Uniport just means one. Antiport means against. So in this case, you'll see that there are molecules moving in opposite directions through the channel across the membrane. This is the second type of system. Lastly, there is coupled transport, where the cell links a uniport and a symport. That is termed coupled transport. Here, the uniport moves something across the membrane against a gradient, and that requires energy. Then, the molecule that was moved across will couple with something else that the cell really wants, and this coupled set of molecules is allowed to flow into the cell without further expenditure of ATP. The second step didn't cost the cell. This is a coupled system. Changing topics from cell membranes and the movement of substances across membranes, we are next exploring the cytoplasm of cells. First, some terminology, some definitions. The cytosol is the liquid portion of the cytoplasm. The cytoplasm includes everything that's inside the cell, the DNA, ribosomes, RNA, everything. Inside the cell membrane is the cytoplasm. The cytosol is the liquid part, the solution of the cytoplasm. Inclusions are the little compartments inside the cell. The cell may store some reserves of some particular substances. Different species of cells may put different things into storage. There are species that put iron to be magnetized to detect the magnetic fields of the earth. Other cells store PHB. Thus, inclusions are a general structure, but different species use them for different types of storage. Endospores are an interesting structure. Not all bacteria have them. The ones that do are able to survive in really harsh conditions. Think of an endospore as like a hibernation scenario. When cells that are capable of making endospores find themselves in an environment that's shifting towards hostile, less water, less nutrients, too much waste, they will start to produce an endospore. The endospore will keep all of their important components safe until things get better. So there's evidence of endospores that apparently have survived many hundreds or thousands of years. They come back to life when they're put in a supportive environment. Not all bacteria are capable of producing endospores. Only some gram-positive bacteria are capable. This is important. Not every bacterial species can make endospores. Only a subset can, which belong within the gram-positive group. Ribosomes are a structure you may be familiar with. They are structures that are important for protein synthesis. We'll revisit them in much more detail in a future chapter. For now, you can remember they play an important role in making protein for the cell. The cytoskeleton consists of fibrils that give the internal space of the cell some organization and a bit of structure. It was thought for many years that bacteria don't have a cytoskeleton. They do. It's just not as complex or as large as what you find in a eukaryotic cell. Bacteria do have a form of a cytoskeleton. Now we're going to be moving into eukaryotic cells and drawing some distinctions between the eukaryotes and the prokaryotes. You might wonder about glycocalyces. Do eukaryotes have glycocalyces on the outside? Well, they might. They're never as organized as a prokaryotic capsule, but when they do have one, they serve the same purpose, to help anchor the cells to a surface or each other and help prevent them from drying out. It gives some resilience to the cell membrane located below. It can even help function in cell-to-cell recognition, but it's nowhere near as organized as prokaryotic capsule, and you don't find it in every type of eukaryotic cell. When it comes to cell walls, some eukaryotes do possess them, but not all. You probably already realize that plants have cell walls, as do fungi. Algae have cell walls as well, and some protozoa do too. There are various polysaccharides that can make up the cell wall. They're linkages or modifications of glucose, as was NAG and NAM of peptidoglycan. Cellulose is the structure of plant cell walls, as well as for some fungi. Fungi might also use chitin, which is a structure found in the exoskeleton of, say, a lobster or insect. Fungi might also have glucomannan, which is another version of a carbohydrate. When considering algal cell walls, you will see that they have a lot of variability too. Sometimes they're made out of cellulose. Sometimes it's different proteins. They might have agar, which is something we use in the laboratory, or carrageenan, which is something that we use in food to smooth out the texture, as in ice cream, for example. Algae can have other types of molecules like calcium carbonate, but basically algal cell walls are very diverse, more diverse than even the fungal cell walls. This electron micrograph is showing a cell wall that's bordering between two cells. The cytoplasmic membrane is the ruffled, darker structures. Then we have cell wall in between. When we're thinking about the cytoplasmic membranes of eukaryotes, they have the same general structure described by the fluid mosaic model. The membranes are constructed of phospholipids, but they also have steroids and cholesterol inserted in the membrane, which helps keep them fluid, meaning flexible, so things can move around. Interestingly, we can have what's known as membrane rafts, meaning when the cell makes something, it inserts it into the membrane from vesicles, and thus it will be in high concentration in an area that's called a raft. It may take a really long time for those components to diffuse and move around on the surface of the cell, so you have these high concentrations of particular components in certain regions of the membrane, and those are called membrane rafts. Of course, the cytoplasmic membrane, just like the cytoplasmic membrane or cell membrane of a prokaryote, is going to control movement of what's coming in and out of the cell. It uses the same general processes that we discussed in the diffusion section. There'll be diffusion, facilitated diffusion, osmosis, active transport. All of those processes are the same. In animal cells, in cells such as amoeba, and in cells like our white blood cells, they can perform endocytosis, a process of membrane movement to bring components into the cell. They also perform exocytosis to export things from the cell. This is an electron micrograph of two cells that are next to each other, a multicellular animal of some sort. You can see these double dark lines on each side of this space. These are the lipid bilayers. The phosphate groups are looking very dark, and the hydrophobic fatty acids is the thinner, lighter area in between. We have an intracellular space between these two cells. The cells might be talking to each other with molecular signals because this space is not very large. The cells could release substances that the other cell would detect. Try to keep this in mind as we move into other parts of the semester where we do talk about cell signals. This image is showing you an amoeba-like cell, or a white blood cell. It is ingesting these little particles that might be a cluster of yeast cells. See the extension coming out? It's called a pseudopodium. The pseudopodium will wrap itself around and essentially fuse together so that the particle will be contained within a membranous container. This vesicle, which can then be fused with another cellular vesicle, will digest the components. This is termed phagocytosis, a process we will go over in much more detail when we get to the immune system later in the semester. Not all eukaryotic cells have flagella. Some do. Some gametes, such as sperm or the equivalent in plants, can have flagella, and some protists move via flagella. When it comes to flagella in eukaryotic cells, they serve the same purpose as on prokaryotes, to propel the cell. However, the structure of a eukaryotic flagella is much more complicated. If you look here, you can see that these structures, if you look here, you can see that there are structures, the pink little rods, called microtubules. They're made up of tubulin protein. They're arranged in these 9+2 arrangements. We have 9 pairs that are around the perimeter, and then we have a central pair. That's referring to the 9+2 designation. The other important difference from prokaryotic flagella is that the entire flagellum is encased within a membrane. The cytoplasmic membrane extends all the way over the whole surface of the eukaryotic flagellum. That's different and not similar to the prokaryotic flagella. The flagella also have to hook into the cell, much like the prokaryotic flagella had that basal body to hook in. The anchor structure is also constructed with microtubules. In this case, the arrangement is a little bit different. The microtubules are arranged as 9+0. There's these triplets out here, and then there's 0 or nothing. No triplet in the center. So the arrangement 9+0 is a little different. Eukaryotic flagella also move a little differently. They don't rotate around, they undulate like a wave. Cilia are really just short flagella, but unlike flagella, where there might be only one or a few, there's many cilia on a single cell. Cilia are strictly a eukaryotic cell structure. You will not find cilia on a prokaryotic cell. When you examine the structure of cilia, they look just like flagella. Just think of them as very, very short flagella. But a cell that has cilia won't just have one. They have many. They may be constrained or confined to a region of the cell, or they may cover the cell, but a cell with cilia will have many. Within the eukaryotic cytoplasm, you will find ribosomes. They function like the prokaryotic ribosomes, but the structure is bigger. There are differences in eukaryotic and prokaryotic ribosomes. More on that difference in a future chapter. Eukaryotes have a cytoskeleton that is much more extensive than prokaryotes. They are made up of different proteins. The microtubules we just discussed as part of the flagella and cilia are also part of the cytoskeleton. The protein that comprises microtubules is tubulin. And you can see the arrangement in this illustration. Microtubules are the larger of the cytoskeleton components. A microfilament is smaller. It's also made of protein called actin. It's the same actin protein that you find in your muscle cells. Here it plays a role in the cytoskeleton. Microfilaments are much smaller than the microtubules, but the intermediate filaments, as the name suggests, are in between the microtubules and microfilaments in size. There are a lot of different types of intermediate filaments, composed of a lot of different types of protein subunits. We are not going to try to name them all here. There are a lot of them, depending on the cell type. In some, the cytoskeleton does give some shape and structure to the cell, but it's also important for organizing the inside of the cell. It helps anchor the organelles where they belong. They play a role in endocytosis and moving amoeba-like cells by forming the pseudopodia. And when organelles stream around in a cell, they do it with the help of a cytoskeleton. In some, the cytoskeleton is more complex in a eukaryote compared to the prokaryote, and it's time to stop. We'll conclude part 3 and pick up with part 4 of cells.